Did you make up that
So this is
mnemonic?
just another cartoon or flow chart
That's pretty good.
that shows myelo and lymphopoiesis. So, how do you go from being a pluripotent stem cell to being a mature eosinophil? So during this hemopoiesis, different cell lines, and this is a cell line, eosinophilic cell line, the basophilic cell line, the neutrophilic cell line, etc. Those are all cell lines. What happens is they'll get a cell surface receptor. They'll develop a cell surface receptor that will bind to a molecule that will tell them that they are going to be an eosinophil or a basophil. Once the receptor is present on any one of these cells, the cell is said to be determined. That means its fate is decided and it's committed to one path to maturity. So what that means is, if you are a pluripotent stem cell, you're going to get a signal to go down this pathway. This guy will make a receptor, and once the receptor is present on this guy, that cell is said to be determined, like he's determined to be an eosinophil. That means its fate, the fate of being an eosinophil, is decided. And that means it's committed to go down one path. So if you get your special eosinophilic receptor here, you can never become a basophil or a neutrophil. You can only become an eosinophil. And this is all just cell differentiation lingo that you have to learn. And then you can have a hormone or a growth factor will bind to the cell receptor, and then once the hormone or growth factor binds to the receptor, it's going to stimulate this process called differentiation. If you are a cell undergoing differentiation, you are maturing. So basically, just vocabulary, I would say, here. You can go online and even, get different, you could Google cell determination, and you could read different ways of saying this. That might make more sense, but this is what I like to say it. So for example, see how I squished in the red blood cell because it wasn't part of the cartoon? You can barely tell, right? But there is this hormone. It's called EPO. Have you heard of it? EPO. They use it for blood doping. That's short for erythropoietin. Erythropoietin is a hormone. If erythropoietin is present, it's going to bind to the erythropoietin receptor on immature red blood cell, and that's going to tell this immature red blood cell to become a mature red blood cell. And we'll see more about we'll talk more about EPO in a second. Maturation then is just development of a mature, specific, aka specialized blood cell type. So on which side of the cartoons are this cartoon are all the blood cells mature? The right side or the left side?
Right.
The right side, yeah. And then there's just things that have to happen to you when you become mature. And that's what I just talked about. It's kind of weird because it's stuff that you don't really see. Like you can talk about the muscles and you just think about the muscles, but this kind of stuff is all kind of invisible. It's cell biology. So a type of hemopoiesis, myeloid hemopoiesis, subtype is erythropoiesis. That's just development of red blood cells. So here's the stem cell. This cell gets its erythropoietin or EPO receptor. That means once it binds erythropoietin, it goes down this pathway of differentiation to become a mature red blood cell. Once the EPO receptor is present, there doesn't have to be anything bound to it. You just have to make a receptor telling yourself that you're going to become a red blood cell. Once the receptor is present, the cell is determined. Once the cell is determined, its fate is decided. The gods have decided its fate, and it won't go off the path. It will become a red blood cell. And then once EPO binds to the receptor, that starts the process of differentiation into a mature blood cell. Again, just cell developed hemopoiesis terminology. Okay. And that's what I'm not gonna ask you these, all these cell names along the process of differentiation. Thrombopoiesis. If you see, oh, it tells you right there. Darn, I was going to ask you what kind of cell type this is. But it's a megakaryocyte. And remember, megakaryocytes, they just start fragmenting chunks of their plasma membrane, and that becomes a platelet. So in the red bone marrow, you have an immature cell that will differentiate into a megakaryocyte. It says here it's a multinucleated cell that releases bits of cytoplasm called platelets. This doesn't look multinucleated. It just looks like, so this is kind of a bad cartoon for this. Multinucleated would mean there's more than one nucleus. In this cartoon, it looks like there's just one giant nucleus. It should be multinucleated. Oh, let me ask you this. See, in the cartoon, that's a white blood cell. You guys should know, what kind of white blood cell is that? Just look at its nucleus. Say it loud, say it proud. A neutrophil. Yes, a neutrophil. Excellent. And then here's just a bunch of stats on how platelets form. And you can read about that if you want to, but I'm not going to touch you on it. All right, get out your cards. Come on, slackers, where's your cards? Brett, where's your card? Why don't you have one? Okay. Why don't you have one next to Brett? Okay, I've got some, I'll give them to you. If you said D, you are correct. You rock it.
I'm getting good at doing educational guesses.
What is the goal of hemopoiesis? How about another H word? Homeostasis. So if, let's say you get a really deep cut and you lose a lot of blood, you've lost a lot of red blood cells. There's a certain number of red blood cells homeostatically that you need in your body to carry oxygen to your tissues. So you've got homeostasis of red blood cells, you need a certain number. If that number goes down, in order to maintain homeostasis, you have to make more red blood cells, and you do. What's the hormone that will allow you to make more red blood cells? EPO, yeah, erythropoietin. And this is just more details that we're, okay. Erythrocyte homeostasis. How can you, or what are some conditions that you might encounter in your life that would decrease your red blood cells? First one, hypoxemia. Well, you guys know this, hypo means below, right? Emia always means blood. So this is just, and well, hypo is low, and then ox would be oxygen, yeah, and emia is blood. So this would be low blood oxygen. One way you can have low blood oxygen is you've got a decreased number of red blood cells. Red blood cells carry oxygen. If you don't have the red blood cells to carry oxygen, you're not carrying oxygen, for the most part. That's called hypoxemia, low red blood cells in blood. Hypoxia, that's low red blood cells or low oxygen to the tissues. So you can have low red blood cells, low oxygen in the blood, low red blood cells, low oxygen in the tissues. In the tissues is hypoxia. Different ways you can get low red blood cells. Hemorrhage, that's the easiest one to think of. You just start bleeding out and you lose blood, you're losing red blood cells. You can also have this problem, hemolysis. I think, is that right? There's some kind of venom that destroys your red blood cells. The venom, it just likes red blood cells. Some snake venom or something, can't remember. But anyway, you can just start hemolyzing your red blood cells. Where have we seen a word like this before? Hemolytic disease of the newborn, HDN, with the Rh negative cartoon, the mom making antibodies to the fetus's red blood cells. Antibodies cause the red blood cells to lyse. If your red blood cells are destroyed, the hemoglobin leaks out, it can't carry oxygen, and you die. No, you don't always die. It depends on how many red blood cells are destroyed. So what are all the H words we have learned so far? Homeostasis, hypoxemia, hypoxia, hemorrhage, hemolysis, and hemolytic disease of the newborn. And what's the process to make blood cells? Hemopoiesis. Yeah, so we got a lot of H words. You might want to make a note of that so you don't get messed up on the test. Hypoxemia. So let's check this out. Where are we? Hypoxemia. So you're here. It could be anything. Let's say you go climb Mount Everest to find Zen because you couldn't find it here at altitude. So you go up to Mount Everest. Lower oxygen pressure, lower oxygen binding to the hemoglobin, and you just have hypoxemia. The liver and kidneys will sense that. The liver and kidneys will make EPO for you. EPO goes to the red bone marrow. What's EPO looking for in the red bone marrow? Why does EPO go to the red bone marrow and not anywhere else?
Stem cells.
Receptor on the stem cells. So you're both right. Receptor on the stem cells. So now your body, if there's a whole bunch of EPO present, you're going to really make red blood cells quickly. That's accelerated erythropoiesis. It's going to increase your red blood cell count up to normal. You'll get increased oxygen transport back into the normal range. And this is a positive or negative homeostasis loop or homeostatic loop. Positive or negative loop?
Negative.
It's a negative because you started with low red blood cells and you ended with high red blood cells to get the red blood cells back into the normal range. That's homeostasis. Homeostasis is you always want to keep stuff in the normal range.
Hypoxemia and hypoxia naturally go together.
Yeah, they do. Yeah, because if you're hypoxemic, you're not getting blood to the tissue, so oxygen to the tissue. Okay. We said decreased atmospheric oxygen is one way to become hypoxemic. You can also be exercising really hard and use up all your oxygen. Well, not all of it, but use up a lot of your oxygen. And that will make you hypoxemic, and emphysema will also make you hypoxemic. Yeah.
How do you tell when somebody is hypoxic?
That sounds like a good clinical question. I mean, are you looking for physical signal?
Yeah.
Well, I would say altitude sickness. You know how people get kind of woozy and they don't really know where they are? That means they're not getting enough oxygen to the brain, so they can't think clearly. So one thing would be a phenotype.
It goes hand in
Where they're
hand with being
disoriented.
cyanotic, too.
What?
It goes hand in hand with being cyanotic too, so you start to turn blue, like your lips. And your
Will you start? Yeah, that's the next thing I was gonna say. If the red blood cells aren't there carrying oxygen, then you will go from having a normal skin color to cyanotic, which is like a bluish color. So those are two things I could think of as a non-MD. But anyone else out there in the clinic? I mean, yeah, I know you work in a clinic, so thank you for that. Cyanotic. Okay. Predict the outcome of an overdose of the hormone EPO. So you have to think about what EPO does. So, what does EPO do? It's looking for an EPO receptor, it finds an EPO receptor in the red bone marrow, what kind of cells? Yeah, baby red blood cells, so, or nascent red blood cells, and it tells the young red blood cells to become mature. So now, if there's a lot of EPO, overdose of EPO, yep, you're gonna crank up your production of red blood cells, that's gonna make your blood very viscous, and now your heart is gonna have to pump syrup
That's true. Yeah.
instead of blood, and you'll have a heart attack or stroke. What response would you expect after traveling to high altitude for 2 weeks? So now you're at altitude, the oxygen pressure is a lot less,
Well, 2 weeks you're supposed to act like that.
there's less oxygen available, so what would your body want to do?
EPO.
Yes, C. Kidneys would make elevated amounts of EPO and you would make more red blood cells. All right, here's a red blood cell, here's a red blood cell. What's this thing? That's a good guess,
Oh,
but
it's sickle
it's
cell.
not a platelet because look how big it is. Yeah, it's a sickle cell. Probably everyone's heard of sickle cell anemia, right? Okay, so this is sickle cell anemia. So what are the different kinds of anemias? There's a whole bunch, but I'm gonna give you the ones that you need to know for the test. So if you're anemic, you could have a decrease in your hemoglobin molecule. Remember, hemoglobin lives in the red blood cells and carries oxygen. Or if you're anemic, you could have just a low number of red blood cells. This is probably the easiest one, like I said, to think about. Hemorrhagic anemia is you're bleeding a lot, you're losing red blood cells. That makes sense, right? Hemorrhagic anemia. Hemolytic anemia, that's where the red blood cells break down. We said an example of that would be hemolytic disease of the newborn. And oh yeah, there is rattlesnake venom, that's it. The rattlesnake venom causes your red blood cells to break down. You could also be anemic because your kidneys are failing, they don't make EPO. If your kidneys don't make EPO, EPO doesn't go to the red bone marrow and tell your body to make red blood cells. If you're undergoing chemotherapy, you could get aplastic anemia. So the chemicals suppress red bone marrow activity. Nutritional anemia. If you have low iron, you need iron, we'll see this in a minute, but you need iron in your hemoglobin molecule to bind oxygen. If iron is not there, your hemoglobin molecule will not bind oxygen. So it's kind of a chemical reason. You need this vitamin B12 to do hemopoiesis. And if you're a vegetarian, you could get pernicious anemia unless you eat a proper vegetarian diet, which I'm not sure what that is. I'm a vegetarian, but I don't even really know how you're supposed to eat it.
Complex
How you're supposed to do it.
carbohydrates.
Meat's too expensive. Just kidding. I have another reason, but it's an even dumber reason. Being a vegetarian. Okay, if you are anemic, these are the consequences. If you've got low red blood cells, instead of your blood being viscous, it's not going to be viscous. So think about your blood. Everybody's seen their blood before. It's a little thicker than water, but it's not really thick. If you're anemic, you don't have red blood cells, it's going to be even thinner than it usually is. Decreased viscosity. You're going to be tired, you're going to be lethargic. That makes sense. You're not getting oxygen to your brain cells as much. You're going to decrease the blood osmolarity because red blood cells contribute to blood osmolarity, that is to say pulling water into the blood. If your blood is less viscous than normal, you're going to decrease the blood resistance to flow. So think about it. Let's think about it this way. If your blood is very viscous, is your blood going to resist flowing very quickly?
Yeah.
If it's very viscous, Yeah, cause it's so thick, it's not gonna flow very quickly at a high rate. If it's really abnormally not viscous, so it's real thin, it's gonna flow real fast. It's gonna decrease resisting blood flow. So if you decrease resistance, you're gonna flow real quick. And if you're anemic, low red blood cells, you're gonna increase the amount of fluid that goes from the blood into the interstitial fluid. So the interstitial fluid is just the fluid between cells. So what that means, if you don't have the red blood cells in your circulatory system to hold fluid in, where's that fluid gonna go? If that fluid, let's say mostly water, is not in your circulatory system, it's gonna go out into the tissues. And then you're gonna, what will you see if you get a bunch of fluid in your tissue? What's that called?
Swelling.
Edema, yeah. So if you are anemic, you're gonna increase the fluid going from your blood to your tissues or your interstitial fluid, and you're gonna see edema. I'm gonna jump over this. I'm gonna go over this quick so you guys done. Why is EPO given to people undergoing kidney dialysis? So if your kidneys aren't working, you're not making EPO, so you gotta give it exogenously. Why would athletes take EPO? Increase the red blood cell and oxygen carrying capacity, yep. If you increase EPO, what happens to the viscosity of the blood?
Increases.
It goes up. What happens to the blood osmolarity? If you increase the number of red blood cells in your blood,
It goes up.
it goes up. And red blood cell production goes up. What happens to your hematocrit? Instead of a normal, what's the normal range for hematocrit? Like 30, what is it, like 35 to 42 for women and higher for men? What's gonna happen to your hematocrit? Is it gonna go up or down?
Up.
Up. Yep. If there's too many red blood cells, that's called polycythemia. So poly means many, cy is cell, emia is blood. Hematocrit can get up to 80%. And we've talked about the blood viscosity. So you can have primary polycythemia, where you have cancer of the erythropoietic line in the red bone marrow. What's cancer? What's just just in general, what's the easiest way to think of cancer? Cells, overproduction of cells. Cells are just out of control. They grow out of control and you get a tumor. That's the easiest way to think about it. If you have primary polycythemia, that means your red blood cells, you're just making a whole bunch of red blood cells in a cancer way. You could have secondary polycythemia. Why would dehydration lead to an increase, a relative increase in the number of red blood cells?
Because you're lowering the amount of water.
Yes, you're lowering the volume of water, so it's relatively, it looks like they're more concentrated, yeah. Relatively, it looks like you have more red blood cells. You can also get secondary polycythemia with hypoxemia stressors like an increase in altitude, we talked about that. And smoking does that too. And vaping and chewing, just kidding, I don't know about vaping and chewing, I have no idea. Sickle cell anemia, so here's a normal hemoglobin molecule. Here's the hemoglobin molecule in sickle cell anemia that allows the hemoglobin molecules inside the red blood cell to clump. And when they clump, it gives it that weird sickle shape. And this is just the genetics. Well, I'm not going to touch on this. This is genetics. Here's the DNA sequence. Here's the amino acid sequence of the protein for hemoglobin. To go from a normal hemoglobin to a sickle hemoglobin, you change these 3 letters in the DNA sequence. That changes this amino acid, glutamine to a valine. And then you get that weird shape for hemoglobin, and you get sickle cells. That's the genetics of it. Here's a red blood cell. This cartoon just shows you a single hemoglobin molecule, but there's literally millions of hemoglobin molecules in each red blood cell. So the chains, the beta chains in hemoglobin, those are all proteins. Inside one of the alpha or the beta chain is this heme group, and that's where the oxygen sits. So adult hemoglobin, you have 4 globin proteins. 1, orange, green, red, and purple, but they're not really that color in real life, just in the cartoon. Within each globin protein, so the green thing is globin, inside there is a heme group. Inside the heme is an iron, and then stuck to an iron is oxygen. Every hemoglobin molecule, healthy hemoglobin molecule, can carry 4 oxygens. Where do they carry the oxygens in this cartoon? In the heme group, yeah, right here. One heme group in the orange, 1 heme group in the green, 1 in the purple, 1 in the red. When you bind the first oxygen, that causes the whole hemoglobin molecule to contort and open up a binding site for the second oxygen. Second oxygen binds, hemoglobin contorts again, opens up binding for the third. Binding of the third causes contortion, opens up binding for the fourth. We'll talk more about this when we do respiratory system and how the gases travel, but a little bit of CO2 can bind to the protein portion, or the globin protein portion in hemoglobin. If you are hemoglobin and your full name is oxyhemoglobin, that means your heme portion is carrying an oxygen molecule in the blood, and you're red. Hemoglobin is red. Deoxyhemoglobin, guess what that is? Without oxygen. It's the heme portion minus oxygen, and the heme molecule is purple. So does that make sense now that your oxygen, or your arteries carry red blood and your veins carry blue blood? Because your veins are carrying deoxygenated blood back to your heart. So your veins are usually blue. But wait, there's more hemoglobins. Carbaminohemoglobin. That's when CO2 is bound to the globin portion. That's called carbaminohemoglobin. And then there's carboxyhemoglobin. That's when the globin portion is carrying carbon monoxide. Carbon monoxide is lethal. So that's what you should get. Don't they have those carbon monoxide detectors for your house?
Yeah.
To see if you're going to die from carbon monoxide? So for some reason, when carbon monoxide and oxygen, they're both competing to bind to hemoglobin, carbon monoxide wins. If carbon monoxide binds to hemoglobin, oxygen won't bind to it. If oxygen won't bind to hemoglobin, you don't get oxygen to your tissues, you don't get oxygen to your brain, and then you die. Oh, I know this one. So, if someone comes into the clinic and their skin is a cherry red, that's a good indication that they have carbon monoxide poisoning. Sometimes students will tell me this stuff because they work in the clinic, and then I remember to write it down. So there you go, future clinicians. Or if you see someone and you go, wow, look at that person, they've got like cherry red skin color. It's not like a sunburn, it's not like embarrassing. Then you'll go, oh yeah, that's right, they probably have carbon monoxide poisoning. Yeah, get them to the hospital. Get them to the hospital. So for you guys that love chemistry, instead of just this orange blob being the heme portion, here's all the atoms and the bonding involved in the heme molecule. All those sweet double bonds, those methyl groups, nitrogen's in there, lots of carbons, carboxyl groups in there, down there. Anyway, that's the heme molecule. There's iron, and that's exactly where oxygen's gonna bind. So this is a test question. I'll have a A, B, C, D, and E, because the test is multiple choice. If all the hemoglobin were free in the plasma versus bound to hemoglobin in red blood cell, what would happen to blood osmolarity and blood pressure?
Decrease.
What do you think?
It decreased.
So let's think about it. If every single red blood cell has like 1,000,000 hemoglobin molecules in it, and now all those red blood cells burst. So let's say you had a system where you had 100 red blood cells. Each one of those red blood cells has 1,000,000 hemoglobins. Now those red blood cells burst open. Now you've got, 100 million hemoglobin molecules where you once had 100 red blood cells. What's going to happen to the blood osmolarity? Is it going to go up or down? It's going to go up. If blood osmolarity goes up, that's going to draw fluid into the blood vessels. What's going to happen to the blood pressure? It's going to go up. Yeah. And then the blood pressure goes up, that high blood pressure is going to tear apart those small blood vessels called capillaries because they're real weak. And then you'll see edema. So you'll have high blood pressure and edema. If you've got some disease like the rattlesnake venom, causes the red blood cells to burst open, all those hemoglobin molecules go out into the blood, osmolarity goes up, blood pressure goes up, and capillaries start to get torn apart. And then here's the answer. It gives you a little bit more detail. So, if osmolarity goes up, then you are said to be, the blood is said to be hyperosmotic. That makes sense because it's hyper. The word hyper almost means too much. Really, we talk all about why this happens when we do blood vessels. I almost feel like I'm kind of not telling you the whole story right now. It makes me feel bad. I just had to confess that. But it would take me a half an hour to really explain this well. So just memorize this and we'll talk about it more later. I know, that's terrible. I gotta change that around. Because I'm imagining that, I'm hoping that you guys really understand osmosis and osmolytes and osmolarity. I don't think that you do that much. Well, let me say this. Maybe this will help. So in your blood right now, you've got particles sitting around, floating around. You've got cells and platelets and antibodies. They're all floating around. They're all osmolytes. Oh, there's proteins in your blood. They're all osmolytes. That means they're particles that are drawing fluid into your blood vessels. Now, if you've got 1,000,000 hemoglobin molecules locked up in a single blood cell, acting as a single osmolyte, now that blood cell bursts, you've just dumped out 1,000,000 more osmolytes that's going to pull in a whole bunch more water into your blood vessels. Anytime you pull water into your blood vessels, that increases your blood pressure. Because you've increased your blood volume, you increase your blood pressure. So think about that.
Is that a permanent situation that happens? Or can it...
No, and then that's a good question because then we'll see with kidney homeostasis, you'll regulate that and you'll urinate out more fluid. Okay, red blood cell, life and death. So we've already kind of looked at this. Erythropoiesis in the red bone marrow. You make the erythrocytes, they circulate in your blood for about 120 days. But at some point, your red blood cells are going to die. They mostly go to die in the spleen. And then in the spleen cells
don't just throw away all the parts of your red blood cell. The spleen will take the hemoglobin and degrade it into heme, that heme portion and the globin portion. The globin was protein, so it's just going to be hydrolyzed free amino acids. You can use those again. The heme's going to save the spleen will save the iron for you, so you can reuse it. But then other parts of the heme moiety, and remember you saw it was huge. It's got a lot of carbons and hydrogens in it. That part will be converted into these subspecies, and then eventually that parts of the heme molecule you'll deposit in your fecal matter. So you have to regulate old red blood cell hemolysis. So red blood cells, when they go to die and they undergo hemolysis, if that's not regulated, those hemoglobin molecules just go out in your blood and cause a whole bunch of problems. So your spleen takes care of that. And it says old red blood cell hemolysis must be regulated or renal failure is possible. So if you have a whole bunch of hemoglobin molecules, they're going to clog up your kidney. And the functional unit of the kidney is called the nephron. The thing that does the filtering in the kidney is called a nephron. And if it gets clogged, then you can undergo renal failure, so your kidneys don't work. And I just said this, if in the spleen, that's where red blood cells release their hemoglobin, they get torn apart, recycled. And then this guy right here, bilirubin, is the most degraded product of the breakdown of heme. And then I say heme is an organic, non-protein moiety. What does it mean to be organic?
It has hydrogen.
It's got carbon.
Carbon.
And hydrogen, actually. It's got both. But, so remember the heme moiety? It's got a whole bunch of carbon in it, so the heme moiety is organic. And also the heme moiety is non-protein. So all these long chains, those are all proteins. They're all amino acids. This is the heme moiety. It's got all these carbons in it. So that's why the heme moiety is organic. Okay, so bilirubin is a breakdown product of heme. And then bilirubin goes into the bile, and then the bile goes out into the feces. And that's the story of red blood cell death and recycling. All right, I'm going to skip over. I'm going to go 1, 2, 3, 4. Looks like I just have to do 4 more slides, have color on them for the test, and then we'll do the PhysioX. We'll do hemostasis, blood vessel healing, and then we'll do that. Oh, another H word. Yay. All right, blood vessel wound healing. So here we are. Here's a blood vessel wall, which is made of endothelial cells and smooth muscle cells. Here's the vessel lumen. What's a lumen? Yeah, the space or the hole. So if the blood vessel is a tube, all that space inside is called the lumen. So here's a normal blood vessel wall. It's got collagen in it. Collagen is a nice protein that helps make your blood vessels strong. Here's normal endothelial cells. Remember, those are just epithelial cells that are found in blood vessels, so you call it endothelium. Normally, the endothelium is releasing this molecule called prostacyclin and nitric acid. And you can see from the cartoon that in a normal endothelium, a normal blood vessel that isn't torn or damaged, the endothelial cells release prostacyclin and nitric acid. And what does prostacyclin and nitric acid do to the platelets, just from the cartoon? Repels them. Because think about it, do you want platelets to just willy-nilly cling to your blood vessel wall?
No.
No, you don't want that. That's bad. But
blood vessel wall, you're gonna expose some of that collagen that was in the wall. The collagen's gonna stick out, and look what happens. Platelets start to stick to that exposed collagen. And you want that, because you wanna heal that broken blood vessel wall. So start making this platelet plug, and these platelets, once they're stuck, they make ADP, and ADP tells more platelets to come to the site, so you can plug up the wound. That's kind of a norm, that's just kind of a general overview. Of course, there's much more biochemistry and cell biology to learn, but that's the gist of it when you damage your blood vessel wall. Any questions? Step one, so let's break it down into steps. How do you heal a broken blood vessel wall? So let's say let's do this. Everybody punch each other really hard in the arm and damage the blood vessel wall. Their arm. Just kidding, don't do that. And I noticed nobody did it. That's good.
You do it.
Yes, all right, I'll just punch myself. Oh, that hurts. So when you do that, even just a mild punch can damage the blood vessel wall, the smaller blood vessels, not the big ones, but the smaller one. You can make little tears, but you don't have to worry about it. You're not going to bleed out because you have this process called hemostasis. So the first thing you do, all right, let's check this out. Here's your blood vessel. Here's the wall. There's endothelial, these are actually the endothelial cells. See, because you see the purple nucleus. This is the wall that the cells sit on. Here's red blood cells and platelets floating in your blood. Here's the vessel injury, and what are these? What's this supposed to be that's exposed from the blood vessel wall?
Collagen.
Collagen. Yeah, exactly, collagen. And look what happens. Normally the collagen is locked up in the wall, but now it's exposed and it's all shredded, and the platelets detect that and they start to stick to it. That's the first thing that happens, and then once the platelets start sticking, the wall is torn, you get this thing called vascular spasm. Everyone's had a muscle spasm, right? That's skeletal muscle spasm or a skeletal muscle twitch. When these blood vessels start spasming, is that going to be the function of what cell type? What cell type will cause the blood vessel wall to spasm or move? Smooth muscle cells. Yeah, good. So it doesn't show it in the cartoon, but there's smooth muscle cells in the blood vessel. They will start to spasm. That's going to cause a constriction. Now, do these two steps make sense? You want to plug up your broken blood vessel wall with platelets. So you want to plug that up. And does it make sense to draw that opening closer together with spasming? Because you don't want a big opening, a big cut in your blood vessel, because the blood will pour out. But if you start to spasm, you're going to make that opening smaller. Less blood goes out. So that's pretty good. That's pretty smart. Vascular spasm, damaged vascular smooth muscle cells start to constrict. When the whole blood vessel constricts, that's called vasoconstriction or vessel constriction. And then the platelets, when they start sticking to the exposed collagen, they'll secrete more vasoconstrictors, e.g. serotonin. Who knew that serotonin, remember that's the happy hormone, the happy drug? Serotonin also can act as a vasoconstrictor. So you don't, if you're taking some sort of serotonin medicine, you wouldn't want to take too much because well, your blood vessels would constrict too much and you wouldn't get blood flow to your tissue and you die. That's my theme for the day. I don't really know if you die or not. I just say that because that wakes people up. If I say die, you'll die. Counter H words. Hemostasis, hemopoiesis, homeostasis, hemoglobin, and a whole bunch more. Step 2, formation of a platelet plug. So in the beginning, the platelets were just starting to stick. Now you have a buttload of platelets, you've got a platelet plug. And that's good because you're plugging up the blood vessel so that more blood doesn't leak out. Okay. And then these platelets are real sticky. So in addition to vasoconstriction, these platelet plugs, once they start to stick to each other, they're gonna kind of cause such a stickiness that that wound is gonna seal up even more. Pulls blood vessel walls together. And this is a positive feedback cycle. Remember at the beginning we talked about negative and positive. This is positive because the more platelets that stick, they call in even more platelets. Those new platelets come in, they call even more platelets into the plug. It's positive feedback. All right, so that was step 2, formation of a platelet plug. Step 3 in hemostasis is clot formation. Have you heard of clotting factors? Or any clotting factor diseases?
Yeah.
Or who was what Russian family was that in the early 20th century? The Russian family, the little boy was a hemophiliac because he didn't have a clotting factor. So if he even fell down on the playground and he got a hematoma or a bruise, he could bleed out and die because he didn't have one of these factors that allowed for clot formation.
Romanovs.
Pardon me?
The Romanovs.
The Romanovs, that's right, yeah, thank you, the Romanovs. Yeah, but they killed, but then the people taking over the Russian government, they killed the whole family anyway, so. They kept the boy alive for a long time, but then they were all massacred. Oh, well. Oh, those Russians. What you guys do with them? Jeez. Just kidding if you're Russian. We'll be our new overlords one day. You and Chinese. Okay, step 3, clot formation. To form a clot, look at all this biochemistry. That you don't have to know all the biochemistry, but these are all the factors that have to get activated until you form this thing called a fibrin polymer. That's the clot. Here's the beginning of clot formation. Buttload of biochemistry. End product, fibrin polymer, aka a clot. So and I've got in red box what you need to know for the test. When you form a clot, you can form a clot by the extrinsic mechanism or the intrinsic mechanism. So let's look at the extrinsic mechanism. If you have damaged perivascular tissue, we know what damage, we know what it is, but what's perivascular? Around the vessels. Perivascular. So if around your blood vessels get damaged, those cells around the blood vessels will release this molecule called thromboplastin, and you have to know the name of this. Thromboplastin binds to factor 7. If calcium is present, you make factor 10. In the intrinsic mechanism to do clot formation, the platelets release factor 12, which converts factor 10 or factor 11 into active factor, which converts this into an active factor, which converts this into an active factor. With calcium and platelet factor 3 present, the intrinsic mechanism can also get you to factor 10. Now, once you're at factor 10, with all these other factors present, you can activate prothrombin into thrombin, and then with an active thrombin, you can take fibrinogen and make fibrinogen into active fibrin. If these other factors are present, you can make your clot, the fibrin polymer. That's the story of the cartoon. So basically, it's just memorizing a bunch of biochemical steps. Genetically, if you are deficient in any of these factors, you could be a hemophiliac. I'm not sure if they know which factors you have to be deficient in.